Welcome to our lesson on monoclonal antibodies and the uses of monoclonal antibodies. The key questions for today are 1. What are monoclonal antibodies? 2. How are monoclonal antibodies produced? 3. How can monoclonal antibodies help to treat diseases? 4. What are other uses of monoclonal antibodies? 5. What are the side effects of using monoclonal antibodies? The key words you will need to know are antigen, lymphocyte, hybridoma, protein, allergic reaction. Monoclonal antibodies are made by white blood cells called lymphocytes. Monoclonal antibodies have scientific and medical uses. They can be used in diagnosis and detection, as well as treatment. Antibodies are produced by lymphocytes and bind to antigens. Antigens are proteins on the surface of cells, pathogens and molecules. The shape of an antibody and the antigen it binds to is complementary. This means they fit together perfectly. For this reason, antibodies can be used to bind with a specific antigen attached to a target cell or molecule. Task: produce a labelled diagram to show how monoclonal antibodies are produced. Monoclonal antibodies are produced by stimulating mouse lymphocytes to produce the required antibody. There are several steps in the process. In the first step, the mouse is injected with an antigen. This stimulates an immune response in the mouse's body. This stage is called immunization. The most lymphocytes will now be dividing and producing antibodies. The antibodies will be complementary to the antigen that was injected. One of these lymphocytes they are producing, the antibody, is removed and combined with a type of tumour cell. This forms a hybridoma cell. The hybridoma cell can divide and make many monoclonal antibodies. All of the monoclonal antibodies are identical. Task: what type of cell produces antibodies? White blood cells called lymphocytes produce antibodies. Extension: why do antibodies bond to a specific antigen? Antigens are usually made out of protein and have a specific shape that the antibody fits exactly. Monoclonal antibodies can be used to treat diseases. They can be bound to radioactive substances, drugs or chemicals, which are then delivered to a target in the body. The target in the body could be a tumour or a site of disease. This means that cancer cells are killed to prevent it from spreading, but healthy tissue is not harmed. Monoclonal antibodies can also be used to detect diseases and monitor the spread of diseases. This can be useful in monitoring diseases such as HIV, cancer and malaria. Another use of monoclonal antibodies for detection is in testing blood for certain chemicals or hormones. This can be used to test for pregnancy. Monoclonal antibodies also have uses in organ transplants, as they can be used to see if the tissue types match. This will make rejection much less likely. Monoclonal antibodies can also be used to locate specific molecules by binding them to a dye or fluorescent substance, and to detect levels of different chemicals in blood samples. The use of monoclonal antibodies is relatively new, so long-term research doesn't exist. However, common side effects include allergic reactions, chills, fever, rashes, feeling sick and breathlessness. Task. Describe 2 uses of monoclonal antibodies. Monoclonal antibodies can be used to detect pregnancy and can be used to treat cancer. Extension. How are monoclonal antibodies used to treat cancer? Monoclonal antibodies can bind with a substance, such as a radioactive substance or a drug, and it will travel in the blood and bind with specific antigens on the cancer cells. The drug or radioactive substance will be directly administered to the cancer and will kill the cancer cells without causing damage to healthy cells.